I think you're muted.
Okay, how about now?
We can hear you.
That's good. Okay, good. The first time I went in, there was nobody there. So I went back out, came back in again. Second time I came in, I could only, it didn't work. So I don't know, something's going on. So what I was saying was I've turned on the iClicker. It has been doing funny, maybe this is it. It has been doing funny things to my computer when I turn on the iClicker app. So hopefully that is not going to be an issue, right? I'll get straight into it. I'll just double check. I hit recording a couple of times. Yeah, we're recording now, so that's good. Okay. And so last time we actually got, let me get to sharing my screen. Share application screen, no. Window. Okie dokie. Hopefully you're seeing that. Now if okay, so hopefully we're good to go there. Okay, we hear and see you, session. Okay, join session. Okay, we're good to go. So last time we actually got to the slide just after this. We were talking about animals and their life cycles. We talked about direct development. Some animals, such as ourselves, you know, we have offspring that look essentially, you know, like mini human beings, basically. But a lot of animals do not. They have larval stages that are incredibly different from their adult stages. Okay, and they have to go through a metamorphosis to change, for example, from this sea star larva into an adult sea star form. Now, one of the benefits of larvae is that they allow dispersal. Okay, so dispersal is the movement away from the parent source or a current habitat. And it's got some benefits that are associated with it. There are risks, but also benefits. And some of the benefits are that you can move to another free habitat if yours is getting too full, disperse to new locations, and also it allows you to interact with other population components, and so it reduces inbreeding. Okay, you're mixing the gene pool. Sessile animals often have larvae. So sessile animals are those that cannot move around, for example, like a sponge attached to a rock. And there are two major larval forms, the trochophore, which is a radially symmetrical larva that occurs in polychaetes and worms and mollusks that we'll talk about in a bit. So here an example of a trochophore larva, radially symmetrical with a cilia band around it. And the other is known as a Nauplius. Okay, this is a Nauplius larva. They have bilateral symmetry, and they occur in, for example, crustaceans. So if you take a plankton sample from Long Island Sound, either at the moment or in the next month or so, there's a lot of larvae being released at that time. The plankton is full of these types of things. You can also get adult dispersal stages. So these larvae are a little bit, you can imagine like you know, a tree setting pollen out, blowing in the wind. These larvae are a little bit like that in the ocean or in water systems. They're dispersed into the water column and then they drift to new habitats. But you can also have dispersal as adult stages. A good example of that is with motile species, so things that are able to move around, such as butterflies and birds. So a butterfly is the adult stage emerging from caterpillars. Caterpillars are fairly stationary in general on the plants that are living, but the butterflies are able to disperse, like monarch butterflies migrating huge distances up and down the country. Some animals are also colonial. So some animals form colonies of genetically identical, physiologically integrated individuals. So here, multicellular individuals reproduce often by asexual means to produce a larger colony. And a great example of this is something like a coral that's made up of individual little polyps that reproduce asexually into lots of them, and they retain, hang about, let me just put my pointer on, if I can find that. Okay, I'm going to get my laser pointer. Here we go. So here, and they retain these little connections between those individuals, so that effectively there's some sort of communication between them. They can share resources and things like that. So they can build up, these individuals can become colonial and build up, massive colonies of coral heads, etc., forming ultimately something like, an entire coral reef that is made up of all these colonial organisms. So an example of a colonial organisms are coral reefs, siphonophores, which are both types of cnidarians, which is the sort of group that jellyfish and things like that are in, and some sea squirts, which are deuterosomes that we'll talk about in a couple of lectures' time. Life also consists of trade-offs, and this is actually essential in evolution of how different traits and different genes arise within populations, because there are costs and benefits to doing different things, and under different circumstances maybe one approach is better than the other, and so the genes that might control one of those traits become selected within the population and vice versa, and that might depend upon what type of species you are, where you live, what environmental conditions you're experiencing at the time, etc. And egg size is a great example of that. So if you imagine an organism that lays an egg, and you have a certain amount of resources. I have this much energy that I can put towards my eggs, okay? Well, that in itself is a decision, because you've got all the energy that you have from eating your food, and first of all, you decided, in inverted commas, that I'm going to invest this amount of it in reproduction as opposed to another amount for, hunting or defending myself and things like that. Amongst that amount of energy that you're using for reproduction, then you might have a decision to make, not a conscious decision, a sort of physiological decision of, well, I've got this amount of energy, shall I put it into small amount into 1,000 eggs, or all of it into one egg? Okay, so, and there are costs and benefits to doing that. A single egg with all the resources is going to be really well provisioned and should do really well because it has all that energy to it. But if there's a very high risk of that one egg being eaten by something, then you're literally putting all your eggs in one basket. You can do that, but there's a good chance that it might be, eaten by something. So maybe it's under certain circumstances, it's better to sort of spread your bets basically, and put a small amount into lots of eggs. Each one of those eggs hatches, and it's not so well provisioned, it's less well developed, but maybe one or two of them are going to survive, okay? And depending upon the conditions, one type of approach might be better than another. So here, if you think about this example with less provisioned eggs, that can also result in how well developed your offspring are when the egg hatches, okay? Certain types of birds have altricial offspring. So an American robin is a good example of that, where those chicks come out, but they can't fend for themselves. They're just like little hungry blobs, basically, that need lots and lots of provisioning from the parents. Versus something like a duck or other types of ground nesting birds, which come out in a precocious state. That means they're coming out well developed and can sort of fend for themselves fairly well from the get-go. Within a few hours, a duck can be, waddling off and actually swimming behind its mother and things like that, as opposed to these guys definitely can't, okay? So trade-offs. There are trade-offs involved in all sorts of physiological and behavioral, etc., decisions that are part of life and are selected upon. Are reptiles precocious? It depends upon the type of reptile, I'm guessing. So I can't tell you offhand, but it would depend upon the type of reptile that that you have, okay, in response to that question. I'm guessing it's very variable across different types. Turtles would be precocious, right? Yes, compared to an American robin, they would be, yes, right. Okay. So what we've been talking about is the animals, basically. This is a phylogeny that we showed last time of all animals. And one of the questions is, well, what is the root of the animal tree? The root of the animal tree provides clues to early animal diversity. See these branches that come off early on, the sponges, so you have a common ancestor leading to sponges, ctenophores, and then this whole other clade here that ultimately leads to these, triploblasts and etc. Okay, which is the most basal group of animals? In other words, which is the most closely related to this common ancestor? Is a question of interest. So the most recent phylogenetic evidence, so based upon genetics, sort of sequencing of genes and genomes, indicates, actually surprisingly to many people, that ctenophores are the basal group here. This is the group of animals that are little comb jellies, okay? In this phylogeny, so choanoflagellates were included. So if you remember, these are not animals, but we discussed at the beginning of last lecture how the fact that when you look at sponges, they have these little flagellated cells called choanocytes that are very similar to these choanoflagellates. And so there's speculation that that's, these gave rise to animals and sponges were basal based upon morphological traits. It seems like from the genetic evidence that actually it's these ctenophores that are the basal group to these other animals here. So what are these ctenophores? They are radially symmetrical diploblasts. So diploblasts, remember, they've got two tissue layers, the endoderm and the ectoderm. Radially symmetrical, that means that you can chop them up like a pie and they're symmetrical around that, although they have actually got some, there is, I don't know how many it divides into, but it's not pure radial symmetry because they have these combs. I think there are 8 of them, yes. So they have jelly-like bodies. They're actually not closely related to jellyfish, although they look very similar in a way to them. One of the differences is that they have these rows of cilia, 8 rows of cilia that come down around the body, and then these long tentacles here. jellyfish do not have those rows of cilia, for example. They're actually very common if you go, down to the seaside just around New York, and you snorkel in sort of calm waters. They're actually, and you look carefully at the surface of the sea, it's quite easy to, or quite common to see these little things. So they capture prey with these long tentacles. There's about 250 species, and they're all marine, and they have sexual reproduction, fertilizing in open water. Okay. So the important point here is that these ctenophores, the silent sea, they're not the same as jellyfish and the cnidarians that we'll talk about a bit later. Let's go through some of these other animal groups, okay? We talked about sponges, okay, and their simplicity, basically. They are loosely organized animals, okay? So they are monoblastic. That means they only have a single tissue layer, as opposed to those with the two tissue layers, such as the ctenophores, and then the triploblastic organisms that we'll talk about later on that have three tissue layers, okay? So they have no distinctive embryonic cell layers, monoblastic. They have no nerves and no symmetry in the majority of them. You do get these, ones that form these cylindrical forms, but many of them are sort of encrusting rocks and things like that. And they do not have, well, they do have some specialized cells, some of them flagellated, some of them not, but not as distinctive as in many other groups. And their sort of structure is held together by these skeletal elements comprising spicules, which are made of different compounds depending upon what type of sponge you are. In the demo sponges and the glass sponges, they have this hydrated silicon dioxide, okay? And when you look at a sort of dried out skeleton of these glass sponges, it's very obvious to see. It's like this fiberglass network when you take away all the sort of tissue layer. But there are also some that have calcium carbonate, so calcareous sponges, such as this one here. And they're mostly filter feeders. I showed a picture at the beginning of the last lecture showing how these flagellated cells pump water into the sponge, and then it comes out the top here. And as they do that pumping, they're filtering microscopic food organisms out of the water using these choanocytes, those flagellated cells that are similar to the non-animal cells. There are a few carnivorous sponges, but that's a real rarity. Most of the marine, just a few freshwater species, and they have both sexual and asexual reproduction. Then there's this other sort of fairly or reasonably basal layer of organisms, the placozoans, which are abundant, but there's very little known about them. In the first page of the chapter, it talks about the discovery of these things in the 1880s, from people with aquaria, and they didn't know what these things were. And very little extra work was done on them until the early 2000s. So it turns out that they're structurally very simple, no mouth, gut, or nervous system or symmetry. But they are actually diploblastic, so they have two tissue layers with the contractile fibers that are sandwiched between those two epithelial or surface layers. And interestingly, they have the smallest genome of any animal, although it's thought that this actually, it seems like that's a derived trait, that they used to have more complex or larger genomes, but then it's simplified. So it's a derived trait. And the reason why so little is known about them is it's incredibly difficult to actually find these in the wild. They weren't found in the wild until the 2000s. And then they're tiny, they're transparent, they're just like really difficult to study, basically. So that's why not much is known about these organisms. But there's their position within the animal phylogeny has been resolved by genetics, basically. That's been the strongest approach with them. Okay, another group of animals that was in that phylogeny that we showed is this these cnidarians. Okay, again a silent C, and they are primarily specialized carnivores. And what they consist of, they include things like jellyfish, not the ctenophores, which are a different group, remember, but also sea anemones, sea pens, which I'll talk about in a bit, corals, and another group called the hydrozoans. So these are diploblastic organisms, 2 tissue layers, filled with this jelly-like stuff called mesoglia, okay, between the 2 endoderm and ectoderm. Their gastrovascular cavity, in other words, their gut, is a blind-ended sac. So this is like a highly simplified version of a jellyfish, basically. And they have a mouth on the bottom here that just enters into this chamber here. So any food goes in, anything that's digested comes out. So it's all coming through the same orifice there. Okay. So a single opening that functions as a mouth and an anus, effectively. And that's used for digestion in that space, absorption across that wall, circulation and gas exchange. So they're getting, you know, oxygen coming in across this wall here, and carbon dioxide being released there, because they're always pumping, which means that water is circulating in and out of those as they're swimming. And it enables them to swim because it acts as a hydrostatic skeleton. If you remember, that's a skeleton that is made up of a fluid, and fluids are mostly incompressible. So when they squeeze around here, it's like a jet that comes out of there. So, you know, every force has got an equal and opposite reaction. So as they squeeze water out there, the jellyfish will move in that direction. Okay. Now, how do they capture food? Well, they use these their tentacles, and what the group are named after is these types of cells that they have, especially in the tentacles, not shown in the simplified version here, called cnidocytes. These are special stinging cells that have these nematocysts inside of them. These are stinging organelles. This is a diagram of them here. So when you touch a jellyfish, what you're doing is you're kind of hitting a little trigger here on some cells that are on the surface that flips open this little trapdoor, and this nematocyst inside is ejected under pressure. See, it's got these barbs on it, and then they have toxins, you know, venom ejected here from the end of that nematocyst as it is averted. So that's how they're stinging and capturing. So these barbs enable them to sort of stick onto things, and the venom allows them to overcome those prey. And it's defensive as well if you're being attacked by something. So it's partly food, but it can be used for defensive purposes as well. Cnidarians have 2 major body forms that they flick between, and in a way that, you know, you might sort of think of it as a caterpillar and a butterfly, where there are 2 major, you know, body forms. They have this as well. Different types of cnidarians might have more emphasis on being in one form than the other, or they can sort of spend equal time or amongst these, depending upon the group that you're in. So most cnidarians have 2 life stages. The medusa form which is this form up here. This is what we think of as a jellyfish form, basically. Okay, it's that swims around and free within the water column. They release eggs and sperm that develop into this planular larva, so a dispersal stage, that settle on the seabed and develop into a polyp. So that's the other form. These are sessile. So the medusae are motile, the polyps are sessile. They're attached to the seabed. And they're essentially, you know, this upside down jellyfish in terms of their form. This was that stylized version of a jellyfish that I showed you. All it is like flipped upside down, so it would be attached to this to the substrate at the top of that jellyfish bit. And so the mouth is now pointing upwards. They have the tentacles pointing upwards, okay, like that. So these are sessile, attached to the substrate, and they have asexual reproduction, so they can bud into new polyps potentially. As I mentioned, some groups suppress one stage in favor of the other. And overall, there are more than 12,000 species of cnidarians with 3 major clades that I'll talk about: the anthozoans, the scythozoans, and hydrozoans. Okay, so we'll move on to look at those forms. So the anthozoans, they're marine, and they consist of sea anemones, sea pens, and corals. Okay, so here's an example of a sea anemone. This is a single individual polyp. Okay, the mouth is up here. These are the stinging tentacles here with the nematocyst stinging parts. Okay, so these are solitary polyps attached to the seabed. Sea pens are very similar in some ways, but different in others. The main thing with these is that they're colonial, and they have one primary polyp that are the, so I'm just looking to think, are the polyps the anchored ones that release the juvenile medusae? Polyps, yes, going back to that. Yeah, so these, just in response to that question, here is a polyp. They release into the water column these little forms here that form into medusae. That's the completion of the cycle. Sorry if that wasn't I didn't state that. But yes, so the polyps are releasing what ultimately become the medusae. So these sea pens are essentially colonial, but they have the first founder anchors in the seabed and grows up from it, and then it buds off lots of different other types of polyps. The upper part is produced with secondary specialized polyps, some of which are specialized in feeding, and they have a sort of circulation, so they can share resources throughout that colony. And they're really pretty, actually. They tend to, you know, occur in soft sediments. So they're buried into the sediment. Sea anemones like this are more typically associated with hard substrates. And then we have the corals that I mentioned earlier on. So this is a close-up of a coral, and you can see that it consists of lots and lots of these small polyps that have, there's been a founder, and then through asexual budding, have produced more and more. Now, corals, one distinct thing about them is that the majority of forms produce a calcium carbonate skeleton, okay, and that's what builds up the actual reef. They also have these things called zooxanthellae, and these are endosymbiotic photosynthetic dinoflagellates, which I think you probably talked about when you were going through dinoflagellates at the beginning of the semester. And these are really important. So coral reefs are primarily restricted to shallow waters that receive sun, because these dinoflagellates are photosynthetic. And there's a symbiosis between the polyp and these zooxanthellae, so that the corals, which tend to live in nutrient-poor waters, they're releasing wastes that are rich in especially nitrogen compounds, which is needed by organisms that are undergoing photosynthesis. they use those nutrients for photosynthesis, and then the benefit the coral polyp gets is that they get some of the results from that photosynthesis, sugars and glycerol, so essential carbohydrates for themselves, basically. So they have this relationship going on. They are vulnerable, however, to increasing carbon dioxide levels in the ocean, because it makes it harder and harder as water conditions become more acidic through carbon dioxide to produce calcium carbonate skeletons. Okay, the more acidic conditions erode that. Also, if the waters are too hot, these zooxanthellae are effectively chucked out of the cell due to stress responses from the coral polyps. Second group of cnidarians is the scyphozoans. So this is essentially what we think of as jellyfish here, such as this lion's mane jellyfish. You can see with the Medusa bell shape up there. These are all the tentacles hanging down there. Actually, this photograph is interesting because there are certain species of fish, such as one called a whiting, for example, that's related to cod, that have mucus on them, a bit like clownfish in a way, that makes them so they can't be stung by a lion's mane jellyfish. So I just noticed those fish within there. So jellyfish are marine, several hundred species, and they, in them, the Medusa stage is the dominant stage. Okay, some of them might have spent some time. What is a zooxanthella? Okay, zooxanthella. I'm just looking at a comment there. It is essentially these photosynthetic dinoflagellates that live inside the tissue of a coral polyp. Okay, so zooxanthellae, think of that as a dinoflagellate. Okay, and as I mentioned earlier on, all that jelly stuff that makes up a jellyfish is made up of this so-called mesoglia. Okay, which is just like a jelly matrix between the two tissue layers. Okay. What's the difference between jellyfish from sea jellies? Oh, nothing. I think sea jellies is probably the more formal name of it, but most people call them jellyfish. So same difference, basically. Okay. And then the third group that we talked about of cnidarians are called hydrozoans. These are typically dominated by the polyp stage, but it is variable. Is the mesoglia to maintain structure? Yes, I think it is, yes. And also so you can have jellyfish fights on the beach, I think. It's sort of, yes, it's basically a sort of structural support. So with hydrozoans, most of them are colonial with interconnected polyps that share continuous gastrovascular cavities. So they can share resources, as we discussed with some of the other ones. And the colonies arise from the so-called planula larval stage that the cnidarians have. So a single planula will land, it will develop into a polyp, and then that will bud into other ones. And there can be specialization of different polyp types. Some feeding types with nematocysts, those stinging parts of the cnidocytes, and defensive ones, and then also some that do not have nematocysts or cnidocytes that are reproductive polyps. Okay. Okay, so I'm going through that a little quickly because we're slightly behind time. Let's see where we are in the grand scheme of things. Like I said, don't get too freaked out about time though, because whatever you get examined on is whatever I tell you. So that won't be an issue. Okay, I'm going to go through a couple of iClicker questions, and let me start this one off here. So I'll give you 30 seconds. Okay. This goes back to some of the things that, okay, so we've just talked about this. So Australia's Great Barrier Reef is made primarily of nematocysts, placozoans, anthozoans, nematocysts, or medusae. Professor, iClicker has given me trouble. Same here. Okay, you know, it kind of gives me trouble as well. I don't know, we'll have to see how it goes. I mean, the one thing I will say about these iClicker questions is, you know, compared to all the extra marks that you get from iClicker and free lecture quizzes, right? That all totals to 15%. iClickers are sort of, I'm guessing, are going to be less important because you do a lot more questions on the pre-quiz test, right? So these are almost like sort of bonus points in a way, but obviously you want to get every point you can. So hopefully you can get it working. Okay, I'm going to close that down. Let's just see what we've got from here. Okay, so most people are saying C. Yes, we literally just talked about anthozoans, right? Anthozoans are the group that corals belong to, and the Great Barrier Reef is made up of corals, right? So it's the anthozoans. Now, anthozoans, it says no response for me. I don't know what to do. We'll go through this, right? And I put in an answer. Yeah. I did. Now, Dr. Spagnoli was saying that the iClicker was causing a thing. Didn't load. Shouldn't be 1% if it should be. Okay. Why does it say 0.5%? Okay, the reason why it says 0.5% is probably because I set this up so that you get, if you just answer and get it, you know, doesn't matter what you say, you get half a point. And if you get it correct, I set it so that you say you get 1 point on top of that. Yeah, that happens a lot. I'll have a look at the things at the end and see. Okay, why I got it correct, but it says no. Okay, so the reason why you got it correct is I haven't gone in and set the mark. What is the correct one yet? Okay, so even if you got it correct, it's just going to say half a mark at the moment because I need to go in afterwards and say that was the correct one. Okay, that's why. Okay, I couldn't click the answers. Yeah, I don't know. We'll talk about this after. Okay. Okay, let's go here. We'll try one more. I couldn't answer two. Okay, don't worry. I'll take this into account. Okay, we'll see if another one works, right? Dr. Spagnoli was saying that when he tried doing multiple questions, that he would get a flood of answers in after I'd shut it down, etc. If you put the wrong answer in accidentally, that's I can't do anything about that, I'm afraid. Okay, this one, iClicker 2. Let's see what happens here. Which statement is true? Nerve systems, so this is going back to something we covered actually last time. Nerve systems evolved independently in some animal groups. Sponges have a loosely coordinated nerve net. The nerve nets of placozoans enable them to move and feed. And protostomes have a nerve net. Deuterostomes have a centralized nervous system. Okay. I'll give you another 10 seconds on that one. Hopefully people are okay. Okay, I've got 105 responses now. They're still coming in. Okay, I'll give you a bit more. Okay, you need to get your answers in now. 200 responses. They're still going up a bit. 203. Okay, we're leveling out there. 205. Okay, I'm going to stop it there. Okay. What do people say? Okay, D. Protostomes have a nerve net. Deuterostomes have a centralized nervous system. Okay, so you need to go back and look at the beginning of this lecture, towards the beginning of this lecture. We had a diagram of where of the phylogeny of animals. Professor, the first question didn't show up in the second one. Okay, there's too many of those to answer right now. It has when we introduce nerves, it's got a picture showing that nerves nerve nets evolved in the comb jellies and then the cnidarians independently. And then a third time evolved in the triploblasts that consist of protostomes and deuterostomes. Okay, they both have a centralized nervous system. Okay. Oh no, I put E because I saw it was the last choice. Okay, well, anyway, so the correct answer is nerve systems evolved independently in some animal groups. It's actually 3 animal groups. Okay, so A is correct in response to, so which one is the right answer? A. 39% of you got it correct. Okay, righty ho. Okay. Right. So next on, hello. Sorry, somebody just said something.
Yeah, is iClicker just based on participation, or is it based on actually getting it right?
So it depends on how whoever's doing it sets it up. I have set up this one so that you get half a point for participation and 1 point extra if you get it correct. Okay, so if you answer a question, I'm giving you half a point no matter what you get, and if you get it correct, you get an extra 1 point. Now I didn't write that on there because but I haven't gone in and marked them yet, so it won't tell you the full amounts. Professor participation, yeah, that was Professor Holtzman. I'm not Professor Holtzman, right? It's different from class to class and professor to professor. The final marks, I haven't gone in and marked them yet. Okay, it's an extra point, right? Yeah, it's okay. That's the way it is. So okay. Right, I've put some summary on this, which essentially you can think of as being revision notes, right? Yes, please make a participation. Well, you get some for participation. I'll give you some for getting it correct as well. Okay, so that is the end of that lecture, okay? And we're going to move on. You may issue for... Yeah, so may have issues for correctness. I may do. I'm going to go and look at it and see how it works, okay? And then we'll figure it out from there. Okay. Next lecture. Next. Okay. So now we're moving on to talk about the protostomes, okay? In this lecture. Let me just close this down or minimize this. Okay, so this is the first part of chapter 30. So question, Melissa, for exams, do you recommend primarily focusing on the lecture slides? Yes. So if you look at the syllabus in answer to that question, the book has a lot of stuff, okay? And it's not always possible to go through everything in the book. So when it comes to lectures, the content of the lecture the content of the exams will be what we cover in the lectures, okay? And sometimes there might be some stuff in the lectures that's not actually in the book as well. Dr. Baker does that a lot, okay? The content of what is going to be in the exams is based upon lecture content, okay? But you'll get more details if you look at the relevant parts in the book as well. Okay, thank you. Okay, so what we're going to do is then in the next couple of lectures talk about the protostomes. So this is an entire phylogeny of animals, and then the two beyond that talk about deuterostomes. Okay. So if you remember, these groups of animals are the triploblastic animals, those with 3 tissue layers: endoderm, ectoderm, and mesoderm. Oops, I'm not sharing my screen. Okay, so it's gone off. Is it? He's not. Okay, let me, I think when I changed slides, it must have stopped thing. Application. Okay, I'm going to share now. There you go. Hopefully that's working. Thank you. Yeah, I think when I changed the PowerPoint, it must have flicked it off. Good. Okay. So what I was saying is that This is a phylogeny of animals, okay? And let me bring up my, so many windows that are going on here, it's difficult to follow what the hell's going on. There's my laser pointer. Okay, we're going to focus in the next couple of lectures on the protostomes, okay? And the two beyond that on the deuterostomes. These are triploblastic animals, endoderm, ectoderm, and mesoderm. And if you remember, this is the, they have bilateral symmetry. They're sometimes referred to in both of these as the bilateria, okay? And in particular on this lecture, we're going to talk about this group here, the Lophotrochozoans, and also we'll mention the arrowworms as well, okay? Next lecture, we'll talk about the ecdysozoans. Okay, the protostomes account for more than half of all described species. And we mentioned last time that there's a sort of general difference between that protostome group and the deuterostome group. So this group here versus this group here. In some of their early developmental stages that you would have learned about in 105, okay? The protostomes, when you have this blaster pore that develops in the embryonic ball of cells, that develops into the mouth. So the protostome means mouth first, and then the anus develops the other end. In the deuterostomes, it's the other way around, okay? So you have this blaster pore that invaginates into the ball of cells, and that develops into the anus. The mouth develops second, so deuterostome, second mouth, okay? So that's the sort of essential difference between these, although there's one or two complications within that we'll mention a bit later on. Another distinction of the protostomes is the way that their nerves develop. So you remember, not all animals have nerves, but the cnidarians do, so jellyfish and corals and things like that. And then deuterostomes do as well. Now we talked about these cnidarians have nerve nets, so do the ctenophores. In protostomes, they have a central nervous system, so highly coordinated. And a key feature of them is that, number one, they have an anterior brain that surrounds the entrance to the digestive tract, okay? So it's a bit like as if we had a brain that went all around our neck or something. And then also their nervous system it's ventral, okay? The ventral nervous system with paired and fused longitudinal nerve cords. So whereas stomes and chordates, we're chordates, we have dorsal nerve cords. So it goes down our back, our dorsal side. The protostomes have it as if their nerve cord is going down their stomach, you know, down their front, basically, the ventral side. So that's another key distinction of the protostomes. So as I mentioned, they're by far the greatest number of described species are from the protostomes. This is a phylogenetic tree of the protostomes here. And it includes groups such as insects, the most described of species, also crustaceans, mollusks, so things like crabs and lobsters, mollusks, things like snails and so on, and annelid worms. And there are two major clades of these protostomes. So clade, if you remember, is a group of animals derived from a common ancestor. They are the Lophotrochozoans and the ecdysozoans. So this includes, we're going to talk about these next time. This is our focus here. Now you'll also notice that there's one sort of weird group, these arrowworms, they're called chetonaths. I'll discuss them in a second. they don't belong to either one of these groups. Okay, they are protostomes, it turns out, although people thought they weren't for a while, but they don't belong to either of these two groups. So the protostome coelom, so remember coelom is a body, are they the outgroup? Yes, we'll talk about that. They're the sort of, they're not the outgroup of protostomes, but they're a sort of a sister group of all the other protostomes that they belong to. So an outgroup of the Lophotrochozoans and the ecdysozoans. So last lecture, we talked about the ancestral lineage of triploblastic animals have a true coelom, a body cavity that, if you remember, is surrounded all the way around that body cavity by mesodermal tissue, okay? That was distinct from some animals that have a pseudocoel, where only the outer layer is covered by mesoderm. And some animals that had no coelom at all, acoelomates. Okay, so the ancestral lineage of triploblastic animals, such as protostomes, are believed to have had a coelom. And traditionally, before genetics and we were able to sequence lots of stuff, people were building phylogenetic trees purely based upon morphological traits. And the coelom, the structure of the coelom, was a key feature that was being used for that. But since the emergence of using genomics for building phylogenetic trees, it turns out you see all these different groups where some have a coelom, some don't, but apparently they're genetically closely related to each other. So in protostomes, it seems like this is one of those cases where they don't all have a full coelom. It seems like some of them have reverted back to those more ancestral conditions. Here is, we're looking across all of the protostomes, and you can see that the coelom, some groups here, like arrowworms, have a coelom. Bryozoans, this group we'll talk about, have a coelom, but others have a pseudocoelom or no coelom at all. And arthropods have hemocoel, which is sort of slightly different. It's part of their sort of loose blood circulation system. So it's only through the genetic methods that this phylogeny has become clarified, because previously it was all based upon things like just coelom structure. So let's look at some general features of Lophotrochozoans. Okay, so this is, like I said, a broad brush general thing. We just talked about how morphometric traits, you can derive them from an ancestral condition, but it's still possible to flick backwards and forwards and do complicated things with those morphometric traits. And so they're not always in themselves the best indicator of your relationships with one another. Nevertheless, some general types of features of Lophotrochozoans, this subgroup, the protostomes. One is that they possess this thing called a lophophore, okay, which is partly what the group is named after. And I'll show some pictures in a second, but it's a ciliated feeding structure that occurs in some groups, the bryozoans, entoprocts, that's these two here, underlined, brachiopods and phoronids. These are all sort of, less well-known groups, basically. And they're all sessile. So this is a feeding structure that can be used by sessile organisms and for filter feeding, essentially. Another one is this trochophore larva that I showed a picture of earlier on in the diagram. It's a free-living ciliated larval form that has radial symmetry. So that's the other part, the lopho and the trochozoan in the name there. Spiral cleavage during embryonic development is a sort of a general feature, but again, not universal. And a lot of them have a worm-like body form, okay? So this lophophore This is a picture of, this is a bryozoan, one of the groups of the Lophotrochozoans. And you can see that they have this structure at the top here, which is the lophophore. It's these tentacle-like things. So bryozoans can oscillate, rotate, and retract their lophophore tentacles. Okay. Did I put? I'm looking for the notes slide. I'm just wondering if I might have put a on the in Achieve. There's a video of that. In fact, I've got a video on the next slide, I think. So this lophophore structure appears to have actually evolved independently at least twice. Or an alternative is some people think that it was actually an ancestral trait in the Lophotrochozoans that was then lost in some modern day groups. So it's uncertain about how, the evolutionary origins of exactly how this arose. But it's quite a complex organism that is used for food collection. So this is ciliated, these tentacles, and also used for gas exchange. So oxygen in, carbon dioxide out. Okay, this video, let's see if this works. I'll leave that to you to play, okay? Because we're also sort of fairly short of time. I've put the link in the comments section of the slide. Oh, here we go. But it basically shows how this thing works. I'll also speed it up a little bit. So this is 1.75 speed just to get through it. So here's the tentacles. They lead down to a mouth, okay, that goes through there. Background music slaps, okay. There's Beethoven, right? So these are ciliated, and in the bryozoans, the cilia move food down, so you can see the beating of those cilia to the mouth, okay? Fleur de lis, yes, it is fleur de lis, yes, for lis. Okay, righty ho. Let's go to the next slide here. Okay. So the other feature that we talked about was this trochophore larva, another thing that occurs in, many of the Lophotrochozoans. Larvae in numerous Lophotrochozoan groups, including mollusks such as snails and clams and things. Annelid worms, that would include lots of worms that live in the ocean, for example. Ribbon worms, which are distinct, we'll talk about those. Bryozoans that we just looked at, and entoprocs that are closely related to these bryozoans. So it's thought to have occurred in ancestral Lophotrochozoans and then lost later in some groups, okay? So something like an earthworm would lose that because they don't live in water, like, a lot of the other worms do, for example. And it's characterized by this band of cilia. The trocho part of the name comes from Greek meaning wheel, and that refers to this band of cilia that occur around the larva, okay? And that can be, when it beats, can move the larva around, but it also is used for feeding currents. So cilia are used for locomotion, but as I mentioned, it also generates feeding currents. Okay. Spiral cleavage, okay, again, something you learned about in Bio 105. So in the early stages of embryonic development, the pattern in which cells divide can be split into radial or spiral in many instances, although there are complicated forms of that as well. Spiral cleavage is typical of the Lophotrochozoans, or at least some groups. This is where, as the cells divide, they sort of go along this oblique axis and sort of forming this whirl if you look at them from above, as opposed to just all being directly stacked on top of one another. And so, I mentioned about how these morphological features had been classically used for generating phylogenies. These were classically referred to as the spiralians, those that had spiral cleavage. But genetic analysis, of course, has come in and messed that all up, that nice and neat system up for everybody. Turns out that spiralians are not an actual true clade. Spiral cleavage perhaps occurred in the ancestral Lophotrochozoans, but it's not known for sure. And then maybe it was lost in some groups later. So it's not, like these other traits, it's not universal of all Lophotrochozoans. It's a general feature. And then we talked about this worm-like body form, okay? Many Lophotrochozoans have a worm-like shape. You can just sort of, if you peruse this phylogeny, these ones here, the annelids are worms, basically. These pheronids are worm-like, quite a few of them have worm-like shapes that with bilateral symmetry. They're part of the bilateria. And they're soft-bodied, many of them, and meaning worm-like means basically they're essentially longer than they are wide. And this is the useful feature if you're a burrowing organism. As I talked about in the last lecture about how the coelom can be used as a hydrostatic skeleton. If you're an elongated worm shape with a hydrostatic skeleton, it's great for burrowing. But there are lots of major exceptions, as in all of these rules. A big one being in the mollusks, okay? That includes, snails and clams and things like that, which are, if you think of a snail, it's not a worm-like thing. An octopus, it's not worm-like, right? So exceptions to the rule. Now, I mentioned earlier on when we we're talking about the protostomes that there are two major groups, the Lophotrochozoans and the ecdysozoans, but also that sort of out group of them, which was the arrowworms. These are referred to as ketonaths. So these are not leucotrophozoans, okay? They were common in the fossil record. They've been around for a long time, at least 500 million years. They're all marine, about 180 species. And although there's not that many species, they're actually very widespread and very common. If you take a plankton sample from, anywhere between the poles and the tropics, chances are you're going to get these in your plankton sample, and they're highly predatory. I mean, they have these bits on the end here that they use for capturing, fish larvae, zooplankton, those larvae that we've showed from the other organisms. So very important members of marine plankton, even though there's not that many species of them. They have three main body regions: a head, a trunk, and a tail region, but with no circulation system. So wastes and nutrients are moved through the coelom by cilia, and gas exchange and excretion by diffusion through the body surface. So they're long and thin and small, so that allows them to get away with that gas across the surface. And they're hermaphrodites, but they produce male and female gametes. Now, the interesting thing about them is that when they were thought not to be protostomes for a while. So classification of these was debated for a long time based upon their morphological traits. They have some traits that can put them in the protostomes and some in the deuterostomes. So they have a blastopore that develops into an anus, like the deuterostomes do, okay, in the embryonic stage. But they have a ventral nerve cord and spiral cleavage, which is more, typical of the protostomes. But genetic evidence puts them with a high degree of confidence within the protostomes. What is uncertain, or more uncertain at the moment, is where exactly they lie within this. The way that this is drawn here in this in this phylogeny from the book is that the Lophotrochozoans and the arrowworms are down a single lineage here, and the arrowworms are split off of them. So in other words, the arrowworms are a sister group of the Lophotrochozoans. But there's a second argument that actually might place them at a further distance away, more closely to the common ancestor, that would put them along this line here, basically, as a sister group to all of the protostomes. So it's uncertain. If you remember, a phylogeny is a hypothesis based upon the best available evidence. So as more things get sequenced and more things get examined, that will probably become more finely resolved over time. So just a quick recap of some of the major points from section 30.1 in the book before we move on to look at separate Lophotrochozoan groups. We're going to, further on in the lecture, go through each one of these groups here, this lecture and the next lecture. So in general, they have shared derived traits, a blastopore that develops into a mouth, unless you're an arrowworm, anterior brain that surrounds the entrance of the digestive tract, and a ventral nervous system, as opposed to ours, which is a dorsal nervous system. Two major groups of protostomes, the Lophotrochozoans and the ecdysozoans, and arrowworms do not belong to either of those, it's thought. And several Lophotrochozoan groups are characterized by the lophophore and the trochophore, okay, but not universal throughout the whole group, okay? Okay, so let's have a look at these Lophotrochozoans, okay? So here we're focusing in on this area of the protostome phylogeny, okay? Some of the groups within this are extremely species rich, okay? Flatworms, if you look at that, according to the book, there's about 30,000 described species of these. These are free-living worms, things like tapeworms and so on as well, that are parasitic. Annelid worms, that includes the polychaetes and the clitellates. So clitellates include things like earthworms and leeches. These polychaetes are generally marine worms. 19,000 species. And then mollusks. This is a sort of, the biggest group here by far. Things like clams and mussels and snails and squid and octopus. Well over 100,000 described species of those, okay? Whereas some of these others, like these phoronids, obscure sort of group, only 10 described species. So a wide range of those. As we've kind of alluded to before, when we're talking about some of those general features, the Lophotrochozoans are extremely morphologically diverse. Okay, this is a table from the book that talks about their coelom structure. So we've already mentioned that. The type of digestive tract that they have, and then circulation system. The main point here is you can see it varies across different groups. Okay, some of them have a true coelom. We were saying we think that's the ancestral condition. Some of them are reverted to a pseudocoel, and some of them have lost the coelom altogether. Some of them have a complete gut. That means that you have a mouth and a separate anus from a single tract, as opposed to a blind gut, which is just a single opening, okay? and some of them have actually lost digestive tracts as well within some of these groups. And then circulation system, either no circulation system, a fully enclosed circulation system, or something that's a bit loosey-goosey between the two, which is what occurs in mollusks. Okay, so very morphologically diverse in terms of not just the way they look, but their internal structure. Can you clarify what you mean by a single opening? So a single opening, we were talking about the jellyfish earlier on, right? Where when we looked at that jellyfish, it just had a single mouth, okay, but no anus, okay? And anything going in, food would come in, but then any digested sort of feces or anything goes out of the same opening, okay? So a single opening. So imagine that you just had a mouth and no anus. That would be a blind gut, right? It's gross. Okay, it might. Yes, it could be. Okay. So we're gonna. That would be a problem. Okay, yeah, it would. Okay, so we're going to talk about some of these groups, right? In most instances, I'll just pull up the phylogeny here just to remind us of where we are in here. So we're going to talk about the bryozoans and the entoprocts. So these are two groups that are, closely related to one another. The bryozoans are referred to as moss animals. There's about 5,000 species. And the entoprocts, which essentially sort of translates to anus inside, and I'll explain that in a second, there's fewer species, about 170, okay? And these are some of the sort of the main differences between them. These are highly stylized pictures of a bryozoan, okay? So we watched that video earlier on already of the bryozoan. So the bryozoans have, when they feed, okay, the mouth is inside this ring of tentacles that they have, that lofer form. And the anus is outside of the tentacles, okay? Now, it's not that well shown here, but it's on, it's not within the inner ring where the mouth is, okay? That's different in the entoprocts, where the anus actually comes out near where the mouth is, within that ring of tentacles, okay? That is what the anus inside part refers to. Another difference is that in the bryozoans, the food particles are carried from the tip to the base of the tentacles. So remember, they feed upon tiny little planktonic particles in the water. They have the ciliated tentacles, and the cilia move the food from the top of the tentacles down to the base towards the mouth. Okay. The opposite occurs with the ectoprocts. The cilia go from this direction to the tip of the tentacles. Now, I'm not sure, but I assume what they must feed then by folding those tentacles in towards the mouth to get that food, I presume. The other is they differ in their coelom structure, okay? The bryozoans have a three-part coelom, whereas the entoprocts have no coelom whatsoever. So there are some, although they look very similar, there are some differences, both genetic and morphological, between them. Now we're doing for time. Okay, 1:30. I'm going to finish this one slide, and then we'll carry on next time, okay? They're also colonial. Remember, we described that it is where they can build up colonies made up of lots of individuals, of these individuals here. And they're sessile. A few solitary species are motile, but that's very unusual. Most of them are colonial, and they're mostly marine, okay? Okay, and we will take up on this slide next time. Like I said, the fact that we haven't finished the Lophotrochozoans, don't get worked up about it. We'll just fit everything in as it fits in, okay? And you'll be examined on whatever we cover. Okay, Why are you somebody else? Okay, great. Thanks very much, guys. I will. Well, you got spring break next week, right? So have a good spring break. Behave yourselves, and I'll see you on the 6th of April. Okay.
Professor.
Thanks very
Thank you. Stay safe, Professor.
Thank you.
Okay, and office hours at 2 today, 2 to 3 if anybody needs to.
Professor, I have a quick question. Is that
Sure.
so on the previous slide, there was like a chart with the differences, and then this same
Right.
picture. Yeah, so I don't know if it's like different wording in the chart, or maybe there's a typo, but for the endoprocs, it says that the anus is inside the tentacles, but on the picture it says the anus is outside the tentacles.
Oh, I probably marked them the wrong way around. Sorry. Yep. I will. Yes, I need to go and switch that around. I'll do that.
Okay, so the chart is that more
So
accurate.
and I've also put, and there's also a typo. I need to go and revise the slide. You're absolutely right. Okay. To get them correct. Okay.
Okay, thank you.
Okay, I will do that.
Hey, Professor, I have a question.
Sure.
Is there any particular advantage for spiral cleavage for these lophotrochozoans? Like, do we know why it was lost in later groups, or are they just diverse?
That's a great question. That's a great question, and I have no idea.
Yeah,
I don't
I'm a
know
little
what
confused about the spiral
What is
cleavage versus.
you're sort of saying? What is the selective advantage? In terms of evolutionary advantage of having one versus the other.
Yeah, because I thought that they would all, have spiral cleavage, but then it's lost. So I'm just trying to, figure out, is there any rhyme or reason,
Well,
or?
some of them develop into a more complex form of cleavage. So there might be some. It's not just those aren't the only two types of cleavage. You can have modifications upon a theme. So I think some of them have become further modified, so they don't look like spiral cleavage necessarily anymore. So I think that's part of what's occurring. But I don't know the details of how every single little group works.
Okay, right.
Okay.
That does make sense. Yeah,
Yeah.
that clears it up for me.
Yeah.
Thank you.
Okay. Great. Thanks very much.
Professor,
Oh, yeah.
hi.